Hi, this is the first biochemistry lecture. Before going through the lecture, I'm gonna to give you an overview about locomotor module for biochemistry. We are going to study carbohydrate metabolism. We study carbohydrate metabolism because we break down or metabolize these carbohydrates into ATP. This ATP is used in the energy requiring processes throughout the body. So carbohydrates are the main source of energy. That's why we study carbohydrate metabolism. Carbohydrate metabolism consists of glucose metabolism, glycogen metabolism, monosaccharides and disaccharides metabolism. The first topic is the glucose metabolism, which consists of subheadings, which are glycolysis, link reaction, TCA cycle, and etc. This lecture's topic is glycolysis. Now let's go through the lecture. First of all, you need to know what does metabolism mean. It is the sum of all chemical reactions that take place in the cell through multi-step pathways. Multi-step, for example, glycolysis, is made of 10 steps. They are not working separately, which means they are dependent on each other. Pathways is a set of enzymatic reactions in which the product of one reaction can serve as a substrate in the second reaction. So, substrate number 1 would form a substrate enzyme complex with enzyme number 1 to give me product number 1. This product will act as substrate number 2 for enzyme number 2 to form enzyme substrate complex number 2. This equals the product of one reaction can serve as a substrate in the second reaction. Okay. These pathways may be catabolic or anabolic. Catabolic means we break down large and complex molecules, for example, proteins, into amino acids. So we break large molecules into smaller and simpler ones, from polysaccharide to a monosaccharide, from lipid to fatty acid. Anabolic pathway, we are going to move in the opposite direction. We form complex products from simple molecules or precursors. For example, the synthesis of glycogen from the glucose. And here is a very important note. All pathways of carbohydrate metabolism aim to maintain the level of glucose in the blood. This equals to homeostasis. We had homeostasis before in the first module in the physiology, if you remember. And this means that the carbohydrate metabolism needs to maintain the level of glucose in the blood to be constant. Of course, this metabolism is regulated and coordinated so that the production of energy or synthesis of end products is going to meet the needs of the cells. Regulatory signals include hormones, neurotransmitters, and the availability of nutrients. For example, the contraction of your muscle needs ATP. If this ATP is depleted, the muscle cannot contract. Glucose regulation in the blood needs insulin. If insulin is depleted, or I have insulin deficiency, which is a hormone, by the way, blood glucose level will not be maintained. So these regulatory signals are very important. Now let's go to the main topic, which is glycolysis. Glycolysis is the hub of carbohydrate metabolism because all sugars can be converted to glucose. And that's why glucose is a very important molecule, and it's very important to maintain glucose level in the blood to be constant. The glycolytic pathways is used by all tissues for oxidation of glucose to provide ATP and intermediates for other metabolic pathways. As I said before, we oxidize or metabolize this oxygen to provide ATP, which will be used by energy requiring processes. Glycolysis have two types: aerobic, which requires oxygen, and anaerobic, which does not need oxygen. Aerobic, of course, occurs in the cells with sufficient oxygen supply and which have the powerhouse or which have mitochondria. In the aerobic glycolysis, pyruvate is the end product, unlike the anaerobic glycolysis, lactate is the end product. Anaerobic glycolysis occurs in the tissues that have no mitochondria, as red blood cells, or in case of hypoxia, no sufficient oxygen. Here the glucose is converted to lactate and not pyruvate. Actually, why we need mitochondria? Mitochondria is the source or is the site for ATC. ATC is required for oxidation of NADH. Without oxidation of NADH, pyruvate will not continue to TCA cycle and so on. To bypass the aerobic pathway, we convert this pyruvate into lactate. We are going to go through all these things next lectures. Glucose is the only product that can be oxidized by the anaerobic oxidation. So, no other carbohydrate can be oxidized by the anaerobic pathway. In anaerobic glycolysis, the NADH does not produce ATP because there is no ATC due to hypoxia or due to the absence of mitochondria in case of the RBC. For glucose to be metabolized, I need to get up or to take up this glucose from the blood. So I need to transport this glucose inside the cell. Glucose cannot diffuse directly into the cell, and instead it enters by one of the two transport systems I'm going to go through now: sodium and ATP independent co-transport system and sodium ATP dependent co-transport system. The first one, from its name, it does not need ATP or sodium, so its passive process does not need energy, and it occurs by facilitated diffusion. Facilitated diffusion means down concentration gradient from the high concentration, which is extracellular, into the low concentration, which is intracellular, requiring no energy. It is mediated of a family of 14 glucose transporter isoforms known as GLUT, Actually, these transporters have a tissue specificity, which means GLUT1 is only found in the red blood cells and the blood-brain barrier. GLUT2 only present in liver, kidney, and pancreatic beta cells. Actually, pancreatic beta cells are the cells that secrete and synthesize the insulin hormone. GLUT3 only present in neurons. GLUT4 only present in skeletal muscle and adipose tissue. They need insulin. What does this mean? Not all GLUT transporters need a signal from insulin. Some of them are insulin independent and the others are insulin dependent. So GLUT4 is insulin dependent. GLUT5, unlike the others, it transports fructose and not glucose. They transport fructose from the intestine to the portal circulation during fasting. Portal circulation from the intestine to the liver. As I mentioned before, this system uses facilitated diffusion, and this is a very important note. Hyperglycemia is the main stimulus of releasing insulin. What does hyperglycemia mean? High blood glucose level. Require insulin. This insulin will act on the GLUT4 and the GLUT2, for example, to cause the liver and skeletal muscles and adipose tissues to take up the extra glucose from the blood to be stored or metabolized. The second system is the sodium ATP-dependent co-transpose system. From its name, ATP-dependent, requiring energy. Sodium, I need to transport both sodium and glucose at once. This means glucose will be transported against its concentration gradient from low extracellular to high intracellular. But the sodium will be transported down its concentration gradient. This occurs in the renal tubules of the kidney. First, blood is filtered through the glomerulus, and here the glucose enters the tubule. If I didn't uptake this glucose, again, this glucose will be lost in the urine. So I need to conserve this glucose. How I'll conserve this glucose? Through sodium ATP-dependent co-transpose system. First of all, the cells of the tubules will go into transport sodium against concentration gradient from the cell to the blood, causing the sodium in the tubules to pass down concentration gradient into the cell, uptaking glucose with it against the concentration gradient. Here the transporter is not GLUT, it is SGLT. It's found in the epithelial cells of the small intestine and the renal tubules. I have discussed why it's present in the renal tubules, and I'll tell you now why it's present in the small intestine. Now I had a meal full of carbohydrates. If I didn't uptake the glucose against concentration gradient, it will not be stored. If I didn't store this glucose in case of fasting, I will find no energy source. So even I have enough glucose, it will still be uptaken by the small intestine epithelial cells. And that's why we cannot use, in case of a small intestine, the first co-transport system, because it depends on diffusion. And now I don't need diffusion, I need active transport. Glycolysis reactions. We'll start with the aerobic glycolysis, where the pyruvate is the end product. There is a net production of 2 ATP molecules per glucose. The first step in the glycolysis, and as I mentioned, the glycolysis is 10 steps. The first of them is glucose phosphorylation. Why should I phosphorylate the glucose molecule? Glucose is highly mobile, so it can leave cells according to concentration. I need to penetrate them, or I need to trap them inside the cells so that they cannot penetrate the cell membranes. In our cell membranes, there is no transporters or carriers for glucose 6-phosphate. So when I phosphorylate the glucose molecules, we trap them as cytosolic glucose 6-phosphate. As we mentioned before, these reactions requires an enzyme. For glucose to be glucose 6-phosphate, we need an enzyme. And here is our enzymes: hexokinase or glucokinase. But what is the difference? Mammals have 4 isozymes of the enzyme hexokinase, 1 to 4. All these enzymes catalyze the same reaction, which is phosphorylation of glucose to glucose 6-phosphate. They would be categorized into 2 groups. The first group consists of hexokinase 1, hexokinase 2, hexokinase 3. The second group would be hexokinase 4 or glucokinase, which is the second name. If there is any difference between these hexokinases, hexokinase is present in all cells except liver cells and pancreas, while the glucokinase is present in liver cells and pancreas. Hexokinase has a low Km, so it has high affinity of glucose. Even if the glucose concentration is very low, the hexokinase will phosphorylate this glucose. Unlike the glucokinase have a low affinity for glucose, which means high Km. Hexokinase allow the phosphorylation of any hexose, but in a much lower rate than the glucose. Glucokinase, from which it got its name, is very specific for glucose only. When we use hexokinase and when we use glucokinase, hexokinase is used to supply the glucose to the tissues even in the presence of low blood glucose concentration because of its high affinity to the glucose. But the glucokinase, which have a high Vmax, this high Vmax allow the glucokinase to sufficiently remove all the extra glucose from the blood following a meal, after eating. Hexokinase is allosterically inhibited by the glucose 6-phosphate, so its product inhibit the enzyme. Let's see here. hexokinase catalyze the phosphorylation of glucose to glucose 6-phosphate. Once the glucose 6-phosphate level increases, it inhibits the hexokinase enzyme, which is a kind of negative feedback. Unlike the glucokinase, glucose 6-phosphate has no effect in the glucokinase enzyme. Instead, the insulin induces the synthesis of the glucokinase. Here is an application. Some people have a genetic mutation in the glucokinase, which means they have a mutation in the gene that produces glucokinase, leading to a disease known as Moody 2, maturity onset diabetes of young type 2. As we mentioned, insulin induces the synthesis of glucokinase. When insulin secretion is not sufficient, glucokinase will not be synthesized and so will not remove the glucose from the blood following a meal, leading to hyperglycemia. And this is the end of our lecture. I hope it was easy for you. Goodbye.